So, hello everybody. Today we will discuss Wnt Catenin pathway. So, a few things that you need to understand that it is very similar to Notch, right? First thing that there is a repressor, which is Groucho, right? So, this Groucho, what it does, it's sitting in the specific domain and the genes are in switched off position. But once the signal, this beta catenin comes in, it removes it, and therefore there is a transcription. Otherwise, it is switched off. So it was very similar in that terms that there was a notch tail, right? Here it is, beta catenin. Why we are referring beta catenin as stable? Because what happens in unstable state that it is phosphorylated, and later on there is proteasomal degradation. So what this complex includes, there is one which is the APC thing. So this APC is scaffold protein holding complex together. Very important. Now there is an axin-like thing which is also a part of this complex. So what happens is that there is another part which is this beta catenin. What happens is that there is CSK1 being recruited, then GSK3 being recruited. So it phosphorylates, and therefore once phosphorylation occurs, it moves it for degradation. Okay, so this is in absence of Wnt. That in absence of Wnt, we don't have beta catenin. Therefore, our target genes are off. But in presence of stable beta catenin, we remove this co-repressor and there is transcription of Wnt target gene. So somehow it's very similar to Notch, right? Howsoever, there is a complex in here. At that point, in that there was Notch tail, and it was only due to cleavage 2 and cleavage 3, right? So now why we are calling it Wnt? Where is Wnt, right? So it's a signal thing that we will discuss now. So you can see that there are two receptors. One is LRP receptor, the other is Frizzle receptor. What will happen in presence of Wnt? It will come and bind and activate Frizzle. Then the LRP is also recruited. This activated Frizzle will recruit dishevelled towards its side, right? It's present in there, but it is inactive form because this is not being phosphorylated. But here it is activated, right? Phosphorylation will be here, right? Now what happens is this dishevelled will pull axin like a magnet. So it will be pulled in. Once it is pulled in, it will go towards LRP, and this LRP is phosphorylated with the help of CSK1 and GSK3. And once axin is pulled out, somehow the complex is destroyed. This APC is also, been out of it, and this APC also goes towards this side. Therefore, now there is no region of either APC or either axin. There is nothing like that. Your beta catenin is free. to bind to Groucho and throw this out. So this is the overall pathway. I hope you enjoyed it. Now, there is one important clinical significance that this APC scaffold. It helps in putting this complex together, in a stable form. But what if this APC is mutated? So there is a very important gene which is called MYC gene. Due to transcription of Wnt target gene, MYC gene is increased, which is important for cell growth and proliferation. But if my APC is destroyed, the beta catenin is stable and free, not degraded. Very important. So if it is not degraded, then what will happen? There will be accumulation of beta catenin in cytoplasm. And this beta catenin will easily move into nucleus and it will accumulate in the nucleus. And therefore, Groucho will be removed simultaneously and therefore MIC concentration is increased. Therefore, when there is no control, what will happen? There will be cancer, right? So that is why APC mutations are very contagious. 80% of human colon cancers. I hope that I have created uh, this simple difference, but that you have to understand that this is contact dependent, delta based, and here is a local mediator. Here there was proteolytic cleavage. Here there is a complex recruitment. You had a notch tail. Here you have unphosphorylated beta catenin because the phosphorylated one is destroyed in proteasomal degradation, right? In general, this notch is waiting in the membrane because there is a heterodimer due to cleavage 1, but cleavage 2 and cleavage 3 is absent because there is no delta and it remains in the plasma membrane, right? So generally default state is waiting at the membrane, but here there is constant degradation of beta catenin, right? These regions are activated. It displaces Groucho. Here it is RBPSUH. Now the diseases is colon cancer, यहाँ पे Alzheimer's. Now the discovery of Wnt. It is originally discovered in Drosophila morphology, wing development, and here it is INT because of viral integration. It was causing breast tumors, and therefore the combination was Wnt. And in humans, it's about 90. Why Wnt are unique? It is because of this covalently attached fatty acid at N-terminal. Therefore, it increases the binding specificity to cell surface. Now, there are certain inhibitors, right? Like there are some proteins, there is a competitive inhibition in terms of that instead of Wnt, something else will bind, right? On LRP, something else will bind. So there won't be any Wnt, right? Therefore, beta catenin will be destroyed and the transcription will remain switched off. But in case of negative feedback, you inhibit the pathway, maybe by inhibiting B-shepherd to shut off the response. That's a normal thing. Other non-canonical pathways are like with the help of Rho family GTPs, that is to coordinate the cell polarization in epithelium. I hope these are certain functions, right? These are kinases. This is your transcription regulator, right? This is LRP, that is LDL related receptor related protein, which is a low density lipoprotein. Frizzled is very similar to G coupled proteins. Thank you.